All right, guys and girls, our last video, and it's on feedback inhibition. So we have to first remember, and I can cover this up if we need to, remember feedback in general. Remember, positive feedback is where something like increases until a goal is met. And negative feedback is bringing something to a target, that steady eddy, okay, meeting that that thing. That is definitely more common. And when we talk about feedback inhibition, it really applies more to negative feedback than anything. But both feedbacks are mostly controlled by enzymes, okay? So let's talk. What is feedback inhibition? Okay, well, feedback inhibition, or what's good about it, is the end product shuts off the pathway. So right there you can see it's a type of negative feedback, because if you produce so much of the end product, then it's got to say, okay, that's enough, kind of holding that steady, okay? And so that's why you're going to see it as like a negative feedback, okay? Why are we saying feedback inhibition here? Because it deals with the inhibition of an enzyme to work. It prevents the wasting of resources. So we're going to look at this enzyme called, well, it's a substance called threonine. Threonine is actually an amino acid. It's an amino acid. Let's say amino acid. And it's a good amino acid. You need to have it in your body, and it does its job. But then the more important thing it does is it eventually forms isoleucine, which actually has a heck of a lot more jobs than threonine does. I'm going to cover this up there. Okay. So how does threonine become isoleucine? Through a metabolic pathway. Threonine binds to the active site of enzyme number 1, which changes threonine to something else, which binds to the active site of enzyme number 2, changes it to something else, binds to the active site of that, and so on and so on and so on, like a domino effect, until finally isoleucine is formed. So threonine goes through 1, 2, 3, 4, 5 changes to get to isoleucine, but it's all enzyme. Enzyme 1, enzyme 2, enzyme 3, enzyme 4, enzyme 5, that are all going to control these steps of the formation of isoleucine. But here's the thing. Once the threonine produces isoleucine, you don't want any more isoleucine. So how do you say, okay, enough? It's like those pens. Do I dare throw them at you again? Like once I have one pen, I don't need any more pens, okay? So how do you make it stop? You said, I got my isoleucine. It's like I got my pen, okay? Isoleucine, okay, once you've made enough of it, here's what it does. It goes up to that first enzyme, and now it binds allosterically, okay, at like not the active site, but somewhere else. And it shuts that off. It shuts the ability of threonine to attach, so it won't continue with that thing. This is feedback inhibition. It is definitely a negative feedback thing because you're trying to maintain the levels of both threonine and isoleucine. Once you produce it, it's like a pen. Once you got a pen, you don't need any more, okay? And thereby shutting down the thing. Now, other things that do this feedback inhibition, 2 other things I want to talk about. First, the production of cholesterol. You might be like, cholesterol? We don't need cholesterol, right? You don't need it because actually your liver, yes, your liver makes cholesterol, all the cholesterol you would ever need. And yes, you do need some. Remember, we need it in our membranes, our cell membranes, to keep them a little loosey-goosey, or else because we're saturated phospholipids and they'd be too stiff and we'd freeze and crack open or something. So we need a little bit of cholesterol to maintain a little loosiness of our membranes, because our membranes are pretty stiff otherwise, which is good because we don't have a cell wall. But anyway, cholesterol we need. And all the cholesterol you ever need can be made by your liver. Yes, you eat cholesterol, and when you eat cholesterol, then the cholesterol gets in your bloodstream, and your liver says, okay, I guess I don't need to do that. And eventually your liver could stop doing that. Okay, and then it's a matter of like having to maintain your cholesterol levels, just like you have to maintain insulin levels and glucagon and all that other stuff. And you don't want to have one more thing to have to like check your blood with or anything. So production of cholesterol, okay, it starts with like the liver, goes through a number of steps, produces the cholesterol. The cholesterol shuts the liver down, the liver enzymes down from producing more. Production of ATP, glucose, right, gets turned into ATP, the production of ATP from glucose. Glucose gets turned into ATP by cellular respiration. Cellular respiration goes through a number of steps to produce that ATP. But here's the thing we never tell you in first year bio. Once you make enough ATPs, then it says, all right, glucose, I'm not going to break any of you down anymore. We got plenty of ATP, so we're done with you, glucose. And that's when it gets shoveled over into your liver, where it's like stored in the form of glycogen, okay, for like later use. So like when you need glucose, yeah, your body will break it down for however much that you need it for, and then you're done. And that's why also sugars and carbs, when you eat all those sugars and carbs, you can break down that glucose. And if you don't exercise, it's fine to eat a lot of sugars and carbs as long as you're going to exercise, because you need that ATP. When you exercise, you use the ATP, it burns off more sugar in your body. Okay, use more ATP, burns off more sugar in your body to make more ATP. Okay, but if you just eat a bunch of sugar and then you sit there on your rear end, okay, then eventually you're going to make some ATP and it's going to say, well, you've made enough. And all that sugar will get stored in your liver first, but any excess gets stored on your thighs and your buttocks and your arms and around your waistline, and you don't want that to happen, okay? So production of ATP from glucose is also a feedback inhibition. And one more time to summarize this up, feedback inhibition is where enzymes that produce an end result, that end result shuts down the pathway. So you have this pathway of enzymes and reactions, right, that are controlled by those enzymes, but the product shuts down the pathway, saying kind of like, we've got enough, shut it down now, okay? And that's feedback inhibition, and that's it for our enzymes, you guys. Make sure you look at this last diagram, which shows the negative and positive feedbacks, and it gives you another diagram of that threonine to isoleucine. This is just another picture. If you don't copy it from my own, this is even better. I gotta admit, this one's better than mine because I don't talk about all the enzymes in between. But this is good. This is that feedback inhibition, okay? And that's it for our videos on enzymes. I hope that helps. Answer those questions on the review sheet. Bye, folks.